dive into the research, I want to briefly explain what SDR is, considering it's probably the first time you've heard of it, and SDR is kind of the main point in this research. Therefore, I want to enlighten you so you can get a better understanding of this presentation. SDR stands for short tandem repeat. They are DNA regions with short repeat units. It means that in the DNA, there are repeated sequences. For example, in the picture shown, letters C, T, and A are repeated several times. SDR is commonly used in forensic investigation for human identification because the number of repeats are highly variable among individuals. So, it is very unlikely for two individuals to have the same SDR profile. SDR is inherited by Mendelian fashion. It can be passed down by the parents to the offspring. Okay, now so let's get into the research about whether or not degrading menstrual blood can be used as evidence by detecting the SDR profile.
Hello to Dr. Kavita and also hello to my fellow classmate. My name is Shahira and I will introduce my part, which is introduction for this research article. The objective of this article consists of two. First is to explore the DNA consistency and detect the SDR profiling from the degrading menstrual blood sample (MBS) as a valid forensic evidence. So the second objective is to investigate the DNA content collected from the very female participant in comparison to peripheral blood sample PBS used as a reference of the sample for each volunteer. DNA profile SDR. Inconsistency in SDR may result in erroneous exclusion and potentially incorrect conclusion, which harming the identification process as well the crime investigation outcomes. The significance of this research. So the first significance is the specific of the crime can be described using the biological material and DNA reconstruction to create extremely detailed and individualized SDR profiling. So the second significance is to provide the linkage between the criminal, victim and the crime scene, and also removing the innocent people who come as a suspect and may be charged of the crime if the incorrectly are followed. So this is resulting that the investigation time can be minimized. So the problem when we use SDR as our forensic evidence, the first problem is a risky cause the possibility destroying the important original material when the evolution technique include many trial and further DNA analysis. And then the second problem is the analysis is too difficult due to low quantity and low quality induced in early degradation of environmental effects. So the example of environmental effects such as temperature, humidity and also the presence of the toxic that affect the preservation of the sample.
Next, we are going to look at the few methods that was used to collect the data and samples and also to conduct the whole experiment. The first method that was used is population and ethical compliance. In this method, 30 participants consisted of 21 to 38 years old healthy women was chosen based on their age, their body mass index, the number of children that they can have, and also based on their menstrual cycle period. The second method discussed on how to collect and also storage the peripheral and menstrual blood samples. For menstrual blood samples, 50 grams of them will be collected on the second day of menstruation and will be collected from the preserved menstrual cotton napkins. It will be collected within the time intervals of 0 hour to 48 hours at 25 degrees Celsius. For peripheral blood samples, 2 ml of them will be collected via the tubes that contain EDTA coagulant. These samples will be treated as the reference samples, and both MBS and PBS samples will be treated within 3 hours after the collection. The third method is SDR profiling, in which the samples will be isolated with KIA and DNA mini kit by following the SPIN protocol. It will be then calculated with the quantifier duo DNA quantification kit, and it will be duplicated at the negative 20 degrees Celsius. In the fourth method, which is the DNA amplification, the samples will be optimized by Paulplex fusion system protocol with the sterilized nucleus-free water and also the model DNA. It will be conducted inside the 96-part Verity thermal cylinder. In the genetic evaluation method, 24 enhanced STR material and also the post-enhanced fragment will be undergoes vortex processing for 15 seconds. 1 microliter of enhanced samples will be inserted into each box that contain the special mixture of Pulplex fusion allele chain and also the special combination of high diformamide and also CC5. A rubber sterilized spectra plate will be centrifuged for 50 seconds and the samples were denatured for 3 minutes at 95 degrees Celsius. It was then will be cooled down for another 3 minutes with ice. The genotype and allele of samples will be separated by using the BI genetic analyzer and will be defined with GeneMapper IDS 1.1. Next, the confirmation of genotype's quality and also the alleles general construct was used by using the electrophoregram review. In the final method of the experiment, which is the data evaluation, the samples will be evaluated with the GeneMapper IDS 3.2 and the output will be integrated into the Excel spreadsheet. The frequencies of genotypes and also allele will be defined by using the centralized gene counting technique, and it will be then introduced in the form of visual heat map by using the software of SigmaTech 3.5.
Now let's see the results. In the aspect of DNA concentration, which is calculated with a quantifiler O dual DNA quantification kit and a real-time PCR 7500 system, it is found that the DNA concentration decreases as more time passes. So, after 48 hours of degradation, the DNA concentration is not as much as the earlier period of degradation, as depicted in the table. Next is STR genotyping. 720 alleles was analyzed in a total amount of 24 STR loci. As seen in the table, the more time passes, the completion of STR decreases. This means that, for example, in 0, 2, and 4 hours of degradation, the STR found in the degrading natural blood is the same as the reference sample, which is the peripheral blood collected from the same person. And at the 20, 22, 24, and 48 hours of degradation, the STR does not match the reference sample.
Hi, I'm Alia, and now we are going to focus on the discussion of the study. STR profiles are used for identifying personalities because they are very different for various people. These markers are biological independent of the DNA source. Lee and colleagues claim that MBS produce a clear methylation profiles that vary from different phases of menstrual cycles and various use methods for data collection. Using RNA or DNA parent extraction, which is used by House and colleagues before, the source of body fluid such as from MBS itself was successfully identified using STR profiling technique. The majority of MBS and vaginal secretion highlighters were discovered to be representative, rigid, respectable, and highly responsive. In addition, compared to most traditional methods for identifying biological fluids, which are mostly based on enzymatic or immunological patterns that can only be attributed to specific enzyme and antigen in the cells, many of these experiments have issues with specificity and extensive characteristics. In the past research, to detect the origins of menstrual blood, the methods used are lattice agglutination test and the diamond assay, which uses the antibodies for fibrinogen decomposed upwards. However, this fails to be useful as being valid and representative compared to STR profiling.
In conclusion, MBA has been shown to be a reliable source in data at the interval between 0 to 48 hours. So the findings suggest that the possibility of precisely detecting MBS source within the 0 and 48 hours, which may be used as a useful and trustworthy evidence for the forensic investigation.